So Dr. Shawn is from the University of Missouri, and he does some fascinating work with functional MRI, and he's going to give us an update on his studies.
Thank you. So thank you for being, I just realized I was so amazed at how the speakers were able to follow the slides, and I realized there's a big screen right here. Sadly, it's bigger than my TV at home. So I am very honored to be here this morning to share with you some of the research that we've been doing in our lab and others looking at PKU in the brain. I'm using the wrong thing to advance the slides. Let's see here. Okay. Yeah, sorry, I got my own laser pointer. I will use that at some point. The brain is a very complex organ. There are many ways to kind of conceptualize it, divide it, classify it. But one of the most basic classifications we can make is between what we call gray matter and what we call white matter. And so what you're looking at here is actually what we call an axial slice of the brain. So we've just kind of sliced off the top of the head. We really didn't do that. It's an MRI. Hey, you guys got to, there's a table over there. I made everybody promise they're going to laugh at every one of my jokes. But they told me I had to raise my hand to tell them when I was telling a joke. So if we look at it, if this image, the gray matter at the level of the cortex, what we think of the brain proper, is located on the lateral surface. I've kind of colored it in here red. From a functional standpoint, we think about this as the tissue where data is processed, data is crunched, okay? That's contrasted with what we call the white matter, which is more kind of centrally located, medially located. It appears in blue here. These are the white matter represents these long distance connections that allow separate brain regions to communicate or transfer information. Okay. If we zoom in on sort of a neuronal level, this is, again, I'm kind of oversimplifying, but this is the idea, is you have the cell bodies, sorry, let me see if I can do this, the cell bodies, and then you have these long axonal projections, and that's what the white matter represents, is these long projections. And these projections are covered in a fatty sheathing called myelin. And myelin is very important because it allows these connections and this communication to be much, much quicker than if it were unmyelinated. So for example, my example is, let's say, maybe you're not as awkward as I am, but if you've ever stubbed your toe in the middle of the night, right, or maybe during the day, again, if you're me, and you hit it and you get that, you have the immediate sense that I stubbed my toe, right? And then it's like 1,000, 2,000, and there's the pain, right? That difference in the timing between you sensing that you stubbed your toe and the actual dull pain coming is the difference between a myelinated pathway and an unmyelinated pathway. So very, what would you say, very sensitive touch is myelinated, but dull pain is not. So can you imagine in the brain how much slower information would be if we didn't have this myelin? So it's very important. And this is just kind of zooming in another graphic. I was bored. And so this gives you the idea. Everybody kind of gets the idea now, hopefully, of the difference between white matter and gray matter. All right. In terms of PKU, the past focus in terms of neurology research has really been on this white matter. And that makes sense. It's reasonable. I mean, we do see a definite increase in the risk for white matter injury in individuals with PKU. And just to kind of illustrate this, what you're looking at here again is an MRI. Again, we have an axial slice. The parameters of the MRI are tweaked a little bit, so things that were gray appear white and white appear gray. If you look at, on the left, you see an individual without PKU. On the right, you see an individual with PKU with this kind of white matter injury. Now, in the middle of the brain, and I'll see if I can point to these, sorry. You see these kind of white spots here? They're meant to be there. Okay, so the white, way over here. In the middle, those are the ventricles. They're full of cerebral spinal fluid. They're meant to be bright white. But if you look at the scan from the individual with PKU, if you look just more laterally or adjacent to it, see how there's also some more whiteness there? You can see that. I think I have arrows. Yeah, the arrows are pointing to it. That's the type of white matter injury that we can see with patients with PKU. Okay. Now, The good news is that we're learning more and more about the different factors that can moderate the extent of this white matter injury. So one that we've known for a while is timing of treatment is better. Early identified and early treated PKU is associated with less white matter damage than untreated PKU. Right? In addition, adherence to treatment is better. And when I say treatment, I'm talking about Phe level in this case. This is from a study we did a couple years ago. On the y-axis here, you have white matter integrity. It's actually something called mean diffusivity. Don't worry about that. Just think higher is better. Fair enough? All right, on your y-axis, you have Phe levels. And so we can all see this nice relationship between Phe levels and white matter integrity, so that higher Phe levels are associated with lower integrity. Okay. More recently, was lucky enough to be involved in a project that was headed by Desiree White and colleagues at Washington University in her lab, looking at whether there were other factors related to Phe. So is it just average Phe level, or is it something to do with variability in Phe? And so to kind of orient you, this top picture I just put up here, where it says mean exposure, all the different white matter tracts in the brain are colored. Okay, that's what you're seeing in the different colors. The regions that are yellow, those are regions that showed as Phe level went up, integrity went down. And so as you can see, it's much of the brain, right? What's interesting is this bottom image down here. Now that's looking at the relationship with something called standard deviation, which is just a measure of how variable Phe levels are. And what you see again here is all the regions that are in yellow are regions that showed where you saw increased variability in Phe was associated with decreased white matter integrity. So what we're learning is that it's not just about Phe levels, it's also about that variability in Phe. That's important. Okay? So that's what we know about white matter. But what about gray matter? Well, it turns out we don't know a whole heck of a lot when it comes to humans. Looking back, there's been maybe 2, maybe 3 studies done looking at the effects on cortical, so that brain proper, cortical gray matter in humans. And they've been done a number of years ago. And so in that time, between then and now, there's been incredible advances in imaging, in processing, data processing, scientific manners. And so we took up this kind of charge and said, let's go back and look at this, and let's look at it using the newest technology and newest things. So this brings us to our most recent research. And so what we do is, you can notice that this picture of the brain I have up there is much, much simpler than the ones that we've seen before, right? Well, this is what we need to get to give it to our computer so that they're happy to process the numbers. What we do is we have a team of graduate and undergraduate students that work for about 1.5 years going through these MRIs, imagine slice by slice, 1 millimeter at a time, and actually using a big graphical tablet and a computer and delineating what was brain and what was things like skull or dura, and also distinguishing that gray and white matter boundary. So, but it was okay because I gave them credit, and I also gave them the password to my Netflix account. So they would sit there with 2 screens, drawing on one and listening to Arrested Development episodes on the other one. So they were happy. No, they did great. We couldn't have done this work without that team. So once we got down to this, now we could feed it to a computer program, and you'd get an output that looks like this. And so to kind of orient you, what the computer has done is it's gone around the surface of the brain, and it's calculated how thick the gray matter is. All right? And in this image, thicker is green and thinner is blue. So imagine we do this for every slice of every brain, and then we take this information and we reconstruct it into a 3-dimensional representation of the brain. And in this case, it may look a little weird, and that's because, as we all know, the brain is very convoluted, right? It has a lot of folds. Well, what we do is we treat it like a balloon and we blow it up so we can see everything. So what you're seeing is sort of a slightly inflated version. Now we do this for all the brains, and then we use general landmarks. So everybody tends to have these major kind of valleys or sulci in the same places. So we align all the brains, so when we're comparing one brain region, we're really comparing the same thing across individuals. So we did this, and we did it in a sample of MRIs, about 20 from individuals with PKU, 20 without PKU. The age range was fairly large. It was 9 to 33, but we made sure if we had a 9-year-old with PKU, we got a 9-year-old without PKU, and so on. So we matched up on age. We really took everybody that we had available. And here's what we found, is we found that on average, the individuals with PKU showed decreased gray matter volume, or that thickness, in a number of brain regions. And you kind of see them up here. I apologize if anybody's colorblind, but they're all tend to be in more posterior parts of the brain. Parts of the brain that are responsible for things like complex visual processing, recognition of objects, visual attention, just general attention as well. So to be honest, this is the part I love about science, is I got this. I remember I found this, and then I don't know if Christine called me. I think you called me for something else, and she's like, "How's that project going?" And I'm like, "Yeah." So, because what we had hypothesized was actually that if we saw differences, they were going to be in the prefrontal cortex, because probably as you've learned in talking, I talk a lot about the prefrontal cortex. I talk a lot about executive function, executive control. So my focus is usually in the front of the brain. So then we find stuff in the back. And so you called me, I think I just did it. I'm like, I told her, and she's like, "What do you think it means?" And I'm like, "I don't know, call me tomorrow." Fortunately, she didn't call me the next day. But what we started thinking about it, or I started thinking about it, and it turns out, it's not maybe as surprising as we might have expected. I mentioned the white matter injury or abnormalities that we see. When we see those, they tend to start posteriorly, and with increased severity or intensity, they go anterior. So those tend to be more posterior as well. It brings up a lot of questions, right? So one thing is different does not necessarily mean bad. So we don't know if this is a good thing or a bad thing. We also don't know how it correlates with behavior, because that's the most important part, right? Does it matter? That's where our next step in this research goes. Well, again, kind of staying on this theme of we go where the data leads us, and I love that approach. It drives my grad students crazy sometimes, because we'll have a plan, and then the data will come out, and we'll be like, "Well, let's do this," and they're like, "But you said we were doing this." And I'm like, "Welcome to science." So they're great. So in doing the literature review for that study, I actually came across an older animal study where they were looking at the effects of PKU in the mouse model on the brain. And what caught me on that is that buried in this paper, they looked at not only the effects on the cortex, which is the brain proper, but also something called the cerebellum. And in that paper, buried in that paper, I realized that they found a bigger effect on the cerebellum than they did on the cortex. And so for those of you that aren't brain scientists or aren't as familiar with the cerebellum, cerebellum is this little brain located at the base of the cortex, and we do call it the little brain. That's a technical term. It's at the base of the brainstem. It's evolutionarily, it's a much, much older part of the brain. Historically, we know that the cerebellum plays a very important role in motor learning and motor control. But like anything else, as we study it more, we realize it's not that simple of a story. It's actually been implicated in higher abilities like language and executive function. See, I knew I was going to get executive function into one of my talks. There it was. So we decided, we were seeing this in animals. Nobody had looked at it in humans, and as it related to PKU. So we went in and did much the same kind of white matter, gray matter segmentation that I showed you with the cortex, but we did it on a much smaller scale for these cerebellums. We again had about 20 patients with and without PKU. I will note that this is a completely different set of individuals, so it's not the same brain MRIs. It's actually a different set of MRIs. And what we found was interesting is, and I should note, we're not looking at the integrity of the white matter on this one. We're just looking at how much there is. Because it's so small, it's hard to look at integrity. What was surprising is we found no difference on white matter. But where we did see a difference is in that gray matter again. We saw a small but significant decrease in the amount of gray matter in the cerebellum in the patient population versus the individuals without. So again, this is kind of leading us back to say, okay, what does this mean? So we're hoping to do more studies and start looking at motor learning and planning as something that may be an indicator as to severity. So my take-home message, 2 years ago I wanted to sell shirts that say white matter matters. That didn't take off so well. Although I've come to find there's this website, it's not Etsy, whatever it's called, that like everybody sells everything. Etsy, oh my God, it's like a cult. Yes, my house has been Etsy'd. So if you got those shirts, which nobody did, on the back it's going to say, but not the whole story. Because we were focusing so much on white matter, and it is an important part of the story, but there's so much more. There's so much we don't know. I talked earlier to some folks, and they kept asking questions, and I kept ending with, I don't know. And I said, I just need to get a sign. And then they brought up like Bill Engvall, if anybody knows that comedian with, here's your sign. Here's mine, I don't know. So that's not the whole story. Just to kind of summarize, though, what we do know, or what we found thus far, preliminary findings, we see this decreased gray matter volume in both cortex, primarily posterior, in sort of the visual processing and attentional areas, and also cerebellum, which we think about as motor learning. But this is such small samples, right? We've got 20 people, that's almost nothing. The relationship to Phe levels, blood Phe levels, are unclear. The relationship to brain Phe levels, which Susan Waisbrun in her session was talking about this exciting new method that she's working on to actually get at brain Phe, which would be great. We don't know. And most importantly, we don't know what this means for behavior and cognition. As a researcher, what I think about, and kind of my approach is, let's identify all the differences we see, and then go back through that checklist and see what matters and what doesn't. Right? But the first step is identify the differences, and then figure out what really matters, what doesn't matter. Different does not mean bad. Different means different. So, our immediate goal is to secure funding for more brains. Brains. Sorry, I love that. Anyway, see, I entertain myself so much. You have to understand that. And my graduate students taught me this. Sorry, I have to show you this. Everybody can see me. Anybody know what this is? It's the sign for awkward. So I was sitting in a lab meeting, and my grad student looks at the other one and does this, and I'm like, what is that? And they're like, nothing. And it took them like 6 months to actually tell me that whenever I said something awkward or not funny, and they would do this to each other. So anyway, so if you're ever talking to me and just do this, and I'll get you. So anyway, so our goal is more brains. We've been working on this level of 20 individuals here, 20 individuals there. It's time. We're at a point with this research where we need to go is in the hundreds, not in the 20s. And that's our next goal, is to secure, through NIH, through your support, is to secure that kind of support. Again, it's going to have to come from some place like NIH to advance this research so we really understand what's going on. Future directions, additional future directions. One of the questions that's been bugging me, and I know it bugs all the researchers, it bugs you, is why are some individuals more affected, right? Why can you have two individuals that have effectively the same, dietary history, even possibly the same mutation, and you've got incredibly different outcomes in them? We don't know at this point, but I think it's an important question that we have to put at the top of our list. And then, with all the new exciting treatments that are coming out, it's going to be fascinating to know how much of these kind of atypicalities, irregularities that we see in the brain, how much is reversible, right? Is it completely reversible? Is it partially reversible? What reverses, what doesn't? What's that mean for outcome, quality of life? Really important questions that we want to get at. So, as my German teacher used to say, das ist alles, which means that is it. And she used to also say Mach die Fenster zu, which means close the windows on your way out because it's the last day of class. But we don't have windows. But and one other thing. Did you know the German word for kiosk is kiosk? I got that one right every time on quizzes. So that's all I remember from high school German. So anyway, just to acknowledge all my collaborators, Desiree Wright, Christina Aldridge, Rich Hillman, Don Peck, all the different universities that we worked with. They're great. This research that I just showed you literally would not have been done without your support. PKU Alliance gave us a grant to fund a graduate student that was able to coordinate and oversee all this stuff. It might have, I should say, it might have happened, but it might have taken us 6 or 7 years. But by giving us funding to have a graduate student, somebody that is now vested in PKU research, it was huge. It allowed us to do these 2 studies And again, it might have been 5 or 6 years before we did them otherwise. So that's thanks to you. So thank you for that. So that is Dasa Solis. That is all I have, but I'm happy to take questions if anybody has questions.